Great. I will get started. Good afternoon, everyone. I'm Mingyao Li. I'm the seminar host today. It's my real pleasure to introduce Dr. Guo-Cheng Yuan, our seminar speaker for PBF. Dr. Yuan is a professor in genetics at Mount Sinai. In fact, he just moved there very recently, perhaps in September, only a couple months ago. Before his current appointment, he was an associate professor at Dana-Farber Cancer Institute, Harvard School of Medicine. He was also a principal faculty member at the Harvard Stem Cell Institute. Dr. Yuan received his PhD in mathematics from the University of Maryland, but he transitioned into a computational biologist by doing a postdoc at Harvard before joining Dana-Farber as a faculty member. In the last decade, his group has developed a number of computational methods for analyzing and interpreting large-scale biological data. So notable examples include methods for epigenomics data analysis. His group has developed Haystack, DHMM, and cut and run tools in this area. Can you mute yourself? Thank you. So in addition, Dr. Yuan's group has also worked extensively in single-cell data analysis. His group developed GeneClast and Geoto. And his group has also worked on genome editing. I used one of the tools he developed called CRISPR-Riso, which is very popular for genome editing data analysis. I have read many papers published by Dr. Yuan, and I found them to be very inspiring, especially his most recent work on spatial transcriptomics. I'm very excited that he accepted our invitation, and he's going to share his recent work in this area with us today. So Dr. Yuan, welcome. I will now turn this over to you.
Thank you very much, Mingyao. Thank you for the kind introduction. It's a pleasure to be here virtually. And let me just see if we can, is there any way I can share my screen?
Oh, let me make you a co-host. That might be able to do it. I'll do the same for Mingyao.
Okay, good. Now it works. Just like this. Can you see my screen?
Mm-hmm.
Awesome. All right. So thanks again. So like Mingyao said, our lab just recently moved from Dana-Farber. Oops. Just gonna try to move this slides to Mount Sinai a few months ago. So I'm gonna mainly. Okay. So since this is the first time I'm visiting, I thought I'd be good to give a little bit of. Oops, what's going on? Why is it not moving? Okay, great. Of our lab, these are the members in my lab before I moved, and since I moved here, only one person came with me. So people enjoy the cold weather in Boston rather than move to New York. So so as a result, we are kind of building our group. So we are, if people are interested in doing the graduate students, postdocs, software engineers, we have job openings, just so you know. So just to briefly introduce a little bit of the kind of main themes in our research. So the kind of we work in the interface between these the biology and the technology and the computation. And in the biology side, what we really interested in the questions are the cellular heterogeneity, the things that we are mostly interested in, which I will talk about more in depth in today's talk. But also, we're also interested in this mechanism underlying how the genes are regulated in a cell-specific manner, and how that's being perturbed in the human diseases, which I'm not going to talk about today because of the time. So I'm really believer of the new technology, because a lot of these new development in the biology cannot happen without these new technologies. So these are some of the technologies that are we have touched upon over the past decade also. And the more so, we are focusing on the single cell RNA-seq analysis, and also the spatial transcriptomics analysis, which I will talk about today. And one of the main things that we can contribute to the, why am I keep having trouble? To the field is we develop the computational tools for the people trying to analyze these data and try to get biological insights of that. So some of these tools are already mentioned by Mingyao, and the main tools I will talk about today is the Jiaotong. Right? So for today's audience, we probably don't need to motivate why the single cell analysis is important. Right? So, but briefly, in our every human or any animals, the basic unit is the cell, right? So every single cell, they actually behave differently, and they also live in a very complicated tissue environment, right? So if you just use the traditional way to do this, bulk-seq or bulk-chip-seq, whatever have you, you only get an average pattern of that, which is hardly representative of any of the cells in this tissue, right? So for 5 years ago, I mean, I guess the technology developed 10 years ago, but really the momentum started 4 or 5 years ago, which is the single cell RNA-seq technologies, which allows you to isolate individual cells from the tissue and identify their transcriptome. Right? So using this kind of approach, it's started to get a complete map of the different sort of cell types and cell states in the tissue, and really get an understanding like how these cells can switch from, let's say, one, say, undifferentiated cell states to differentiated states, and so on. Right? So we have a lot of information just by doing these single cell sequencing-based analysis, right? So let me just digress a little bit, and along these lines, I want to just briefly mention one thing that our lab has made some contribution, which is a tool to identify these rare cell types from the single cell RNA. We call it the gene class, right? So basically, the motivation is that now we have typically have thousands or sometimes even millions of the cells that are generated by these single cell RNA-seq. Now people begin to wonder, are there any cell types that people really don't know before, right? And these are the cell types that typically exist in the rare frequency. So that's having the power to sequence it's a lot of not large number of cells can give us the potential to identify the cell types. But then the how to identify these cell types is actually not as simple as you would imagine, because as you know, one of the first tasks to identify these cell types is to identify the features that are associated with these cell types. Right? So this is a little bit of the chickens and egg issue. And traditionally, the issue is solved by looking at the overall variation of these gene expression and identify the genes that are most variable. So a lot of commonly used software uses this approach. But this approach is useful for identify these common cell types, but it's really not very useful for identifying the cell type, rare cells, just because that these genes that are expressed only in the rare cells really contribute very little in the overall variability. So to overcome this problem, then we use a different statistic called the Gini index, which is originally used for the socioeconomic study, to identify the genes that are really specific to a small cell types. And if you look at this kind of, roughly speaking, you order the cells for each of the gene, you order the cells from the lowest abundance to the high abundance, and on the y-axis, you look at how many the transcripts are expressed by these cells that you have counted so far. So if the gene is expressed in equally expressed in every single cell, then you would get this diagonal. But if this gene is expressed only in small number of the cells, then the curve looks like that. Therefore, the area between these two curves give a good measurement for the rarity of the when the gene is expressed. So this, the method was first published in 2016. We call it the Gini class. So the reason I want to mention this is because since then, we have made a number of improvement. One of the improvement that we made a couple years ago was to combine this Gini class algorithm together with the traditional, high variable gene based clustering algorithm. And using the ensemble approach, we can identify both commonly and rare cell types. So if you use the original version, you can only detect the rare cell types, but you miss the common cell types. So by using this highly variable genes, we can pick up the common gene cell types as well. So more recently, we made another improvement of this, not in terms of the methodologies, but in terms of the speed. Right? So I think we improve the speed of orders of magnitude, so that now you can start to look at identify the rare cells from say millions of cells. Right? So one thing that just as a quick note, I want to mention that it's not so easy to visualize both the common cells and the rare cells in the same plot, because the typical way of looking at these things using t-SNE or UMAP and so on is based upon the fact that you already know which genes to look at. Because we're looking at different gene features using the rare cells and the common cells, we have to look at multiple views to get a better idea, right? Such as this. And this is the tools that are that's available here, and really welcome you to try it out if you're interested. So now let's go back to this kind of main topic of today, which is that I mentioned that the single cell RNA-seq, as powerful it is, it's quite limited. The main limitation is that it's only confined you to look at the cell intrinsic dimensions, whereas the spatial dimension is actually missed here. And in the past few years, a number of technologies have been developed, such as the seq-fish, MERFISH, and more recently the 10x Visium, SliceSeq, and a number of other technologies have been developed And taken together, what it does is that to provide you not just the intrinsic dimension, but also the spatial dimension as well, to give you like a more complete understanding of the cellular heterogeneity within a tissue, which is really, really important for us to understand, for example, how these tissue environments impact the behavior of the individual cells, even though the cells may come from the exactly same cell state. So as I said, the technology has moving very, very fast, and just in a few years, there's a lot of technologies that have been developed. And then people start to use these technologies to get the biological insights. One of the big things that are a lot of groups, including us, involved in are to use these technologies to generate this the cell atlas, right? In the human different tissues, sometimes in mouse. And the goal is to provide a kind of reference map so that later on when people study the diseases, they can compare to say what is the changes and so on. Right? So obviously the next step of doing this kind of research is to move from these normal tissues to the disease tissues. And I think that there are a number of the studies already been published to demonstrate the value of the spatial transcriptomics. Right? So given this kind of the bigger picture, then one thing that we recognize is that there's still a bottleneck in that there's very few computational tools that actually allows you to do this comprehensive analysis, which is our group taken the initiative since 2 years ago. I tried to develop these comprehensive toolbox called the Jiaotou. And this work is involves a lot of people in our group. As you can see, these are the people that contributed to this work. And but it's primarily led by very talented postdocs, Ruben Dries and Chen Zhu. So Ruben has just recently started his own lab in Boston University, and he's continued working on this area. So just to give you kind of overview of what the tool looks like, we show you a schematic. So you started with this sort of tissue, you generate this data. As I mentioned, there's a number of different ways to generate the data. The technologies are all different. Each of that requires different pre-processing and normalization and very, very detailed methods. But we want to build a generalized in the pipeline so that anybody can use it. So this is why we decided to choose the starting point, not as a raw data, but as a processed data, which encompass these gene by cell matrix. So that's just, you can also get that information from the single cell RNA-seq. If you're familiar doing that. But in one additional component here is the location of the individual cells. And that can come from in the form of cell segmentation method. And sometimes it's come from these kind of the spot locations if you're working on the 10x VCM data. So it can, the exact the meaning of these data can vary quite a bit, but we try to build a kind of uniform pipeline to process these different data. So the most basic element of this pipeline is to do the clustering, right? And this step is not that different than what you would typically do with a single cell RNA-seq analysis, for example, because then you basically do this the clustering, and then you identify these groups of the cells with similar gene expression patterns, and then you look up the biological literatures to do the cell type annotations. So one thing that's a little bit different here is that if you have data such as the 10x Bayesian, for example, or slicing, these are the data that do not have the single cell resolution, then it will be not so much meaningful to talk about the cell type associated with a specific spot, right? Because each spot has multiple cells. So then we, to overcome that problem, we built this tool using this spatial enrichment analysis and to identify the spatial distribution of the different cell types by ingesting also the known gene signature associated with the certain cell types, right? So I will talk about how that works a little bit later. And then the next step is that we're really interested in to know this spatial structures, right? So that's the most distinct pattern of the this type of data. And for that, we have a number of the tools that allow you to do this analysis. First, we can search for the spatial patterns of individual genes or the assembled, the whole, the cell states. We have multiple tools to do that. And in addition to that, we want to ask that how the cells behavior, meaning that the transcriptomic profile is dependent on its cell neighbors, right? So how the cell-cell interactions with each other, right? So, and then lastly, we have a tool that's visualized this data. We call this the Giotto viewer. Okay, so next, just to quickly show you this where you can find the detailed information. We have bioarchive that's published in July this year. If you read an earlier version of the preprint, I suggest to do it again because it has changed quite a lot since the original version. And the tool itself is available in this main website and also in this development website. All right, so let me just dive into this the tools. So for the sake of time, I'm mainly talking about what the what the tool can do, rather than talking about the methods. But I'll be happy to talk about the details of methods separately. So so first, let's talk about this the clustering, and this is I don't need to explain this too much. I just want to show you an example. So the data sets that we apply to is the SIGFISH plus mouse cortex data generated by Locke's lab, and this is a single cell resolution spatial transcriptomic data, which covers about 10,000 genes, and this is a really powerful technologies. And we have the data in the mouse cortex for about 1,000 cells. Oh, I should, by the way, I should mention that the Giotto that I'm talking about today also has the links of a number of the different data sets that we analyzed. And also they give you the procedures, how do we analyze that, so that if you have a new data of that similar kind, then you can use that as a guide to do your own analysis, because obviously every data set requires a different kinds of analysis procedures. For example, some of them only make sense if you have a whole genome data, but then if you have proteomic data, let's only have 40, 50 genes, then it would not make sense. Something like that. So I think that's a really could be useful guide. Anyway, so if we apply the clustering method to this SIGFISH plus data sets, then we can identify these 12 cell types, and so these are the marker genes. As you typically see, we can visualize the data in the UMAP or TSNE if you like, or we can also look at the data in the XY physical space like that, right? As you can see, some of the cell types, cells from the similar cell types, they are co-localized, together, have this kind of layer patterns. Right? So we can also do this in three-dimensional data sets. So this is the merfish data that's generated by Xiaowei Zhuang's lab. So basically what they do is that they have 3D brain tissue and they cut it into multiple thin slices. And by piecing together these information from different slices, you can get a three-dimensional view of the tissue. So this can be done by using Giotto. We can also look at this by individual slices as well at multiple different the resolutions as well. So a little bit things are a little bit more interesting is to look at the VCM data, right? So I mentioned that this is new technology that's widely accessible because that's commercialized by 10x Genomics. Problem with this VCM data is that it does not have a single cell resolution. Each of the spot here is 55 microns, so which is roughly speaking 5 to 10 cells. So how do you get the cell type distribution information from this data that does not have the single cell resolution? So to solve this problem, we use the external gene expression signature that can be identified from, let's say, a single cell RNA-seq. So then we can basically look at the signature genes that are associated of each of the cell type and use these tools that are commonly used for the gene set enrichment analysis, right? So such as the page and rank, just to look at where this gene set is enriched, and from that we can estimate the spatial distributions of different the cell types. Right? So to test if this idea actually works, we actually generated a simulated data, but the simulated data is based on the real SeqFish plus data. So because we want this to be as close to the reality as possible. So the way we generate the simulated data is this: we take a grid, which is about 50 micron on each dimension, just to kind of mimic the size of the VCM, and then we take the gene expression profiles that falling within each of the grid, and then we take the average, and that's our so-called the VCM data. And we apply our enrichment methods to analyze this coarse-grained the data sets, and using this external single-cell RNA-seq data generated by the Allen Brain Institute to guide the enrichment analysis, right? So in this case, we know the truth, right? So because this is the truth comes from the single cell resolution data, and this is the pattern. The color represented the frequency of that particular cell type in each of the spot, right? So that's what this blue means. That's the truth. And the red, that's our prediction, right? So that's the enrichment score, right? Notice that there's a high correlation between our predicted enrichment score based on the prediction pattern and the real data. So this is I think it is quite good. I'm pretty happy that it's also able to detect in this case where even the majority of the cells come from the cell type. We are not fooled by that. We are still able to predict that they are highly enriched in almost every grid. So so that's kind of convinced us it's not a bad method at all. So just as a simple benchmarking, we compare some other simpler methods, hypergeometric-based enrichment methods, or simple, correlation-based methods. And these methods are not as good as the one that are that we use now. So that's why we decided to use those methods. Oh, so I also have to mention that these methods are also robust with respect to these the read depth, which can be a problem if the data quality is not that good. All right, so then we apply this to these the VCM data. This is the VCM brain data. So these are the cell types that are we found to be enriched in these different areas. And I also marked these Allen Brain Atlas anatomic annotations and labeled where these each of the cell type are supposed to be located according to prior biological knowledge. And you can see that a lot of these areas have a pretty good match with the prior knowledge. Again, suggesting that the enrichment analysis is useful. So that's we're doing this. The next time we move on to the spatial patterns, right? So there's a lot of the methods have been developed in the I mean, there are a number of the methods have been developed in the spatial patterns, and I will tell you a little bit of how we approach it. So, so you start with this spatial network. So the edges represented which cells they are next to each other, right? So if you look at the genes with the expression level, some of them they could be randomly distributed, and some of them they can be localized. So we are interested in finding these genes that are spatially coherent. How do we do that? So there are these methods such as the spatial DE and transseq that do this. But we develop our own method called the BeanSpec, and I would later explain a little bit that our method do have some advantages compared to these existing methods. So actually the method is quite simple, right? So this is just schematically, this shows how it works. So imagine that you have these cells with these expression levels of these particular gene. So the first thing we do is that to binarize the gene expression pattern so that the highly expressed genes got called 1, and low expressed, highly expressed cells, we call it 1, and low expressed cells, we call it 0. And then we counted how many the edges that are connecting this these cells with a 0, 1 or 1 and 1. So we create a table like that. So suppose a gene that's highly localized like so, then you would find that these edges tend to connect the cells that are both are highly expressed, right? And sometimes they are both lowly expressed. On the other hand, you don't see many cells at the edges that are connecting one cell that's low expressed and one cell that's highly expressed. So if you look at this contingency table, you would expect that there's a majority of the edges are along the diagonal, and very few edges are in the off diagonal. And this bias can be statistically evaluated by using this Fisher-Exact test. So then that p-value can be used to generate a rank. So then that's how we detect these differential, not differential, sorry, the spatial coherent genes. So then we want to benchmark these methods, and then we have a little bit of problem. Actually, I'll be really happy to hear your thoughts about this, how to solve this problem. but the way that the problem is that what do you mean exactly by a pattern, right? So on one hand, you can say, the pattern is a circle or line or whatever regular shape, but that's clearly artificial, right? In reality, there's no such thing as such a regular pattern. On the other hand, you could have this real situation, but then different people have different views to say, this is a pattern, this is not a pattern, and so on and so forth, right? So there's ambiguity there, right? So how do you solve this problem? And really like to hear your thoughts afterwards if you get a chance to ask questions. So the way we approach it is the following. So we take these, we randomly selected these genes, right? So so this is based on this seqfish data, and we just pick the genes so that the expression levels, they are real, right? So based on the real seqfish measurements, and then as you can see, maybe some of these have patterns, some of them not, but that's not what we're concerned about, right? So here we only look at the spatial, not the spatial, so the overall gene expression distribution, right? That's step number one. And step number two is that we create a spatial pattern, right? And the way we create a pattern is that within that space, we sort of like a defined area, right? So in this case, it's kind of like a rectangular area. We say, this is the pattern, right? We want the genes to be highly expressed over here and not elsewhere, right? And for that, what we could do is that we could start to rank these cells from the low expression to the high expression over here. And then we start to put the highest expressed cells in that area, right? And we do that, randomly, right? So we define this area, and then we pick this, the top expressed cells, right? This for one single gene, and we put these cells randomly here. Whereas the other cells we randomly distributed over here, right? So that's how we do it. And then we want to make the task a little bit more challenging. We say, let's introduce some noise, right? The noise that we introduce is that we allow a probability to randomly switch, right, between the cells in the area, the highly expressed area versus the out, right? So as you can imagine that when the probability is getting like lower and lower, then the data gets more and more noisy. And eventually, it's no difference than just a random gene expression, random spatial pattern. Sorry. So then, with that in mind, we compare these different methods. So there's these the silhouette rank. That's another method that our lab developed, which is not, we don't need to focus too much now. But the one that we care more about is the by BinSpect. We have two variants of the BinSpect version over here. And then we have the three methods that are obtained from literature. So as you can see that if the gene, there's no noise, all the methods are behaving pretty well. And if you have a very, very large noise over here, probability close to zero, then none of the method works well as you would expect. But in the middle, there is a wide range of the probability area where we think that's the BinSpect clearly outperform the other methods. So that's suggesting that these methods, that BinSpect method is indeed useful. But the other advantage is that this is really, really fast. This is all the methods, we apply this to the same data sets. And we can see that our bin-spec method is orders of magnitude faster than the other methods, suggesting that if you have a large data set, then the bin-spec is better. So let me see. Okay, so I need to speed up a little bit. So we apply this method to the real VCM data sets. So this is the kidney. This is the what it looks like. The slide looks like. So using bin-spec, we find these genes. Notice that some of these genes, they have a very localized pattern, and that still can be detected by the bin-spec. And we also can enhance the spatial patterns by grouping the genes that have a similar spatial patterns using the traditional, the clustering. Here the clustering is based on the co-localization of the different genes. And then we indeed find a stronger spatial pattern of the meta gene, averaging the genes expression within each of this module. This pattern is clearer here compared to the individual genes, right? All right, so briefly I want to talk a little bit about the spatial domains. So the method I talked before to you is looking at one gene at a time. But you also want to know in the tissue, are there any areas that are occupied distinct patterns of the cell states, right? So you can imagine that there are some, if you have a cancer tissue, you can imagine there's just areas that are occupied primarily by the cancer cells, whereas some others may have this immune infiltrating cells, right? So you want to identify these regions. So to address that, then we built this hidden Markov random field model, which is published already. So I wouldn't spend too much time talking about it. But bottom line is that we consider the spatial continuity as well as the gene expression patterns of individual cells. For example, here, even though the blue cells has distinct gene expression pattern than the neighbor cells, but just because of this continuity, then we say this is most likely the whole area comes from this one sort of domain versus another one. So this is very different than just doing this the k-means clustering and so on. So this does use the spatial information to do the segmentation. So just to show you quickly, one thing that we observed by applying to a SIGFISH data. So this is a kind of a different data sets that I showed you before. So this is a visual cortex. What you see is that these are the vertical layers, the cortex layers 1, 2, 3, 4, 5, and so on and so forth. And I'm showing the domain structure on the top, and I'm also showing this cell type information at the bottom. As you clearly see that the domain pattern that captures the spatial patterns, the anatomic patterns that's from these different layers, whereas the cell type, you cannot. You look closely, you can see that some of the cells, they are clustered the same cell type, but they have different shapes. Like these guys, they are circular, and these ones are elongated, so forth. These are more elongated. But if you look at the domain patterns, they are assigned to distinct the spatial domains. So these indicate that the really this domain pattern provides the complementary information that can be used to dissect the heterogeneity within one cell types. So that's what we thought, that the combining the spatial information and the cell type information, the two dimensions looking at together, they can give you more information then you can look at the single component alone, right? So, so this is actually quite interesting. So if you have the spatial information, one natural question is that how the cell behavior is influenced by its neighboring cells. To systematically qualify this approach, what we could do is by looking at the expression profiles of a certain gene in one cell type, but neighboring different having different neighbors, and we compare the gene expression patterns in these different contexts. For example, one thing you can do is that to say that we look at this endothelial cells. It can be next to the astrocytes, can be next to this excitatory neurons, inhibitory neurons, and so on. So the cell type specific markers, they have no specificity whatsoever, as you would expect it. But there are certain genes that they are really dependent on these neighbors, and those genes that may suggest a certain cell-cell interactions going on between the cells and neighbors. We can also generalize that into this formally the ligand receptor, the network. In this case, we have not just one cell, but we have a pair of the cells. One look at the expression level of the ligand, the other one we look at the expression level of the receptors. We can do exactly the same analysis. We look at the combined ligand receptor expression level in certain cell type pair context versus the others using permutation test. By looking at the difference of that, then we can evaluate the strength of the dependency of the neighboring the cells, right? So for each pair of the cell types, we can do this, and obviously we can look at it for every single cell type pairs, and looking at every single pair of the ligand receptor pairs. And from that, and then we can start to get a diagram here, whereas the rows here represent each of the pair of the ligand receptor pairs, and each of the column represent a cell type neighbors. And here, this red spots indicate those ligand receptor pairs that are specifically enriched in those cell type neighbors, right? So this is kind of one example to show you that, for example, right? So we have these two the cell types indicated over here, right? So for these, we look at the cell gene expression that when the two cell types are next to each other and the two cell types that are not next to each other, we find that the cell type specific marker genes, they are highly expressed no matter what. But then there are these certain ligand receptor pairs, they are much higher expressed when the two cell types are next to each other, and then they are not, right? So indicating in this case is kind of enhancing interactions. Sometimes we also see the opposite pattern, which we call the inhibiting interactions as well. Both can happen. Okay, so I still have a couple minutes. So let me just, okay, so let me just mention that we also have a visualization tool of this spatial transcriptomic analysis, where you can visualize the same data in the spatial view and also in the gene expression view. We can overlay with other information such as the staining. You can also look at multiple annotations together and try to see where they are differ, and select the cells of interest and save for further analysis. So this just shows that you can also look at not just the cellular resolution, but also at the subcellular resolution. Right. So this module is called the Giotto Viewer, which is a independent module that comes together in this package, right? So just in the last, quickly in the last couple minutes, I want to mention a very recent study we did in collaboration with Long Kai's lab. So, and this is a little bit more forward-looking, right? So far, all I have been talking about is just the RNA, right? So you may wonder, RNA is just the one modality, right? Wouldn't it be nice just to see the RNA and the DNA and the protein all in the same cells all together? And these Long Kai's lab, they're really genius develop this, technology allows us to do just that, right? So I talked to you about the single cell, the sickfish data. So that's the RNA part, but also they have this DNA fish to look at the 3D organization of the chromatin, and they also use this immunofluorescence to tag these proteins. Different proteins. In this case, they're looking at this chromatin regulator proteins, the histone modifications, some of these organelle marker genes, marker proteins, so that we can have a complete, not complete, but complete in the sense that I have 3 different modalities in the same cell. And just to kind of quickly give you like what you can see from this sort of data. For example, one thing you could see is that you can see this sort of high C type of the patterns, where you can have the context between one locus versus another. So here's just a comparison. We are on the top, we're showing the sickfish plus data coming from this study. And the bottom, we compare with high C. You can see that there's a high correlation between these 2 technologies. Furthermore, what they allows you to do is that we can actually looking at the single cell resolution, which is not possible if it's just to do this the high C, right? What also you can do is that you can look at where the location of the individual the proteins, and where get a sense of like how the cell organelles they're organized into the cells. So we are really excited about this kind of data, and we are trying to make sense of the combining all these 3 modalities together, trying to learn new things about the cell states and the mechanism underlying that. All right. So almost the time is up. So let me just summarize. So just the message is very simple. I hope that I convince you that the Giotto, the toolbox, is a useful tool for the spatial transcriptomic analysis. And then I will acknowledge my group listed here, Ruben Dries and Chen Zhu. They are here and here, and the other group members. And this is a wonderful collaboration with Long Kai's lab in Caltech and the funding. And I will put this up again, and hopefully I still have opportunity to answer some questions. Thank you.
Thanks so much for such a wonderful talk. The floor is now open for questions. If you have a question, please feel free to unmute yourself. I saw a question in the chat box. The person is asking, is Giotto compatible with the GeoMX data generated from nanostreams?
It's a very interesting question. So we haven't looked at that yet. I have to be honest with you. But in theory, there's no reason we cannot, right? Because we do have this, I guess in that case, then you would need this in doing this enrichment analysis tools, which could potentially be useful over there. But we haven't tested yet.
Guo-Cheng, I have a question, if I may.
Sure, sure.
I really like the BinSpect algorithm. It's very straightforward, but there's a nice way of measuring what you're looking at. I wonder whether you've tried to look at that at various levels of resolution. So you use the sickfish data where you know what truth is as a way of benchmarking. But you can imagine a situation, let's imagine, for example, that blue cells show up on the left and not on the right, but blue cells never touch each other. They're always surrounded by green cells, astrocytes or whatever.
Right.
There, BinSpect will never pick up the blue association on the left, but if you lower the resolution slightly so that you're looking at clusters of cells, you'll clearly pick up the left-right association. It occurred to me that if you just simulate this data from the seq fish, you might be able to identify an optimal level of resolution. It may actually work better on Visium data than it does on single cell data.
Yeah, that's a great point. Yeah, so I completely agree. So the ultimately you would need some sort of, as you suggested, I think we're getting at is that you probably need some sort of maybe. I mean, you phrase it as a parameter tuning sort of problem, if I understand correctly. But you can also think of that as a multi-scale problem as well, right?
Right. Exactly. And I mean, I would say probably the best strategy to do this would simply be to look at a variety of different resolutions and pick those that give you the maximum signal. It's just, you know, for this particular case where you have single cell resolution data, you could simulate that very easily with the seq fish data. And my prediction is that you'd actually get better identification of sub-tissue clusters at you know, something like 50 micron resolution than you do at 10 micron resolution.
Right, right, I agree. I mean, just a clarification that, I mean, I agree completely what you said. Just a clarification that we're doing this on the gene by gene basis.
Right.
So.
Yep. Yeah. No, I you know, I understand that, and of course that's another benefit potentially in that if these are indeed cell type specific responses, you would expect to get reinforcing effects. You get a better signal for all genes that are associated with one.
That's right. Yep. Yep. Thank you. Yep.
Anyway, thanks a lot. Very stimulating.
Thank you so much.
Any other questions from the audience?
So is there?
I have a
yeah. Yeah, go ahead.
Yeah. So thanks for the really nice talk. I was wondering if you know, in your experience with these different kinds of spatial data, you know, what's, you know, how prevalent are the batch effects in these? Because, you know, in single cell RNA-seq and the sequencing type data sets, you know, we really have to work hard to get rid of the batch effects. How about, you know, for these different kinds of spatial technologies? Have you seen strong batch effects? Like, say, if you know, and how would you even assess?
Right. Yeah, that's a good question. So we have not systematically looked at the batch effects in this context yet. The I mean, I wouldn't be surprised if this kind of thing, you know, exists, right? So what I can say is that at least from a personal experience, the kind of I need to explain a little bit. So the data that they show you, they actually are from, I mean, in a way you can view that from different batches. Let me explain a little bit. So that's basically, it's a stage of 16 or 20, you know, like you know, multiple views, right? So of the data, and you can you know approximately saying that each of the view perhaps is is a batch, right? So in that regard, we for the first the seq fish data, we don't think the batch effect has a bigger role, just because that we are able to identify these the layered structures which get across multiple views. The boundary has no effect whatsoever. in defining this domain. So I thought that that's probably an indication that it's okay. For the SIGFISH plus data, I cannot say that as confidently, because in that case, unfortunately, the viewpoints align with the layer structure. So I don't know to what extent that the difference is because of the batch effect that you talk about, because the real biological differences from the from one layer to another layers. But I wouldn't be surprised, as I said, I wouldn't be surprised that this kind of the issue will be come up in this type of analysis. I mean, the one thing that you have to keep in mind is that one of the source, I mean, at least in my understanding, is that one of the source of having these batch effects for single cell RNA-seq is that you have different efficiency in the application, the reverse transcription. That factor does not exist in the the SIGFISH type of analysis or MORPHISH type of analysis. So yeah.
Thank you.
Sure. Thank you.
I actually have a related question.
Sure.
In one of your slides, you had showed the example. You had the data from that MORPHISH paper. They generated cells from multiple tissue slices from mouse brain. So in that data set, you can actually perform three-dimensional data analysis, potentially identifying genes that have a spatial variation in the 3D space. I was wondering, when you perform the spatial domain analysis and the spatially variable gene detection analysis, how do you analyze data like this that has a 3D?
Right. So the in theory, there is no problem. I should say. So one thing I should mention that the Hidden Markov random field model is based on the network structure, right? So we only look at the cell-cell proximity, regardless of they come from the 2D or 3D, it doesn't matter. So which is different than the traditional Hidden Markov random field model, which is based on the strictly on the grid. So that's I don't think we need to worry about that too much. For that particular data set you mentioned, one thing that you need to consider is that the different slices, they are actually not that close. So it's really, there's no much information you can learn from the neighboring layers because there's a multiple cells, in between. So you have to analyze each of the slides independently.
I see. Thank you.
Sure, thank you.
It was a beautiful work and talk. So I have one question about the convolution of pixels that you mentioned in technologies like Vision, where you have below single cell resolution, so you have multiple single cells in each pixel. So you mentioned about estimating the proportions, right, of each cell type within each pixel. I was wondering if you can go beyond and estimate the actual number of cells in each pixel of each cell type. Is that something?
So that's you could do that if you make more assumptions, right? So which I mean, you would probably have to assume that the different cells approximately express the same number of the transcripts, right? Because otherwise you don't know if any difference is because you have twice as many cells or twice as many transcripts per cell, right? So that's something that, I mean, if you make that assumption, then I guess you could. But actually for VCM, you don't actually need to do that, I guess. There is a staining, HNE staining in the spot, which could give you information about the total number of the cells. We have not used that yet. right? To be honest with you, we have not done that yet. But suppose you use that information, you could potentially answer the question you just asked.
Okay, so you would say based on the total number of cells that are in the pixel, based on the staining, multiply by the inferred proportion, basically.
Something like that. Okay. Something like that. We haven't tried it yet, but I mean.
Okay, very good. Thank you.
Thank you.
Guo-Cheng, I have a related question to deconvolution and enrichment score you presented. I wonder, have you compared the enrichment score analysis with results obtained from cell type deconvolution?
Right. So we did actually. So how do I say that? Actually, we already have a method that's kind of close to finish, I think. But I mean, it's so the short answer is that we find that the enrichment analysis, they both have some sort of advantages, disadvantages, right? So the advantage of the enrichment analysis is that you do that cell type by cell type. So if you have a similar cell types, it's really not an issue here. All right, so you can say, this is not exactly the subtype A versus subtype B. You don't really know that, but in a way, you have that's what you expect, right? So
if you have a very rare cell type in the spot, do you think you are able to pick that out?
Yes, we can. Yes. So well, I mean, rare. What do you mean by rare? So the deconvolution, this done is spot by spot, right? So even though it's rare in the whole tissue, right? In that fragment, in that particular spot, it's not that rare, right? So maybe like if you have 5 cells in that spot, you still have 20%, right? So the transcriptome coming from that cell type, so it's not that rare, right?
So do you see you see a positive correlation of your enrichment score and the cell type proportions?
Yes, absolutely, absolutely. So as I said, I didn't finish, but if you have the cell types that are quite distinct, right, and then the deconvolution results actually match quite well with the enrichment score analysis, and of course the benefit of that is that you have more quantitative information than the enrichment score, right? So. But then the assumption is that the gene expression signature, they are distinct enough for us to do this sort of.
What if you have closely related cell types? For
that's what I mean. So it's probably not work. I don't know. I mean, you guys can try your music and I see if that works, right?
So it doesn't perform very well in this context.
Have you tried that? I mean, I guess I don't know. I mean, we tried the DWS, which we did, and we have that problem. But I don't know. Maybe if you use your method, maybe it's better. But I'll be really interested to hear that.
Yeah, methods like music will suffer from multicollinearity.
Exactly.
So if you have closely related cell types, methods like those will have a difficult time to infer whether the cells are coming from this type or from the other type. Do you encounter similar issues when you calculate the enrichment?
Absolutely. Yeah, absolutely. So then that's why, like, when we have like similar cell types, we tend to group them in a bigger group for the deconvolution analysis, right? Mm-hmm. If you're not deconvolution, that's fine, right? You don't really care. So if you have like a very similar cell type, you get a similar enrichment score, so which is fine.
Thank you.
Yeah, thank you. It's a good question.
Any other questions from the audience? We are already running out of time. Okay. If not, let's thank our speaker again. Thank you so much for a wonderful talk.
All right, thank you for the nice questions and thank you for the invitation. Right, it's a pleasure. All right, take care.
Bye, everyone.